Welcome dear students. Today we will understand about cells. In this session we will just touch the tip of the iceberg to understand basics of the cell biology, which will place us in a position to understand in depth about what cells are and how do they function. We'll cover all of these various topics, starting from the history of the cell theory, the differences of prokaryotic and eukaryotic cells, plant and animal cells, the structure and functions of the various organelles, and also particularly about the nucleus and the DNA. In the part of our first video, we will talk about introduction to cells. There is a huge diversity of cells around us. Many of them are visible to us, many of them are invisible to us. But you might be thinking which cell is actually visible. The cell as such is not visible to us in most of the scenario, but the structures that they form, the tissues or the organs are visible to us. And there are organisms which are only made up of a single cell as well, such as the unicellular organism, including the bacteria, archaea, and protozoa, and even some fungi and algae. However, there are also examples of organisms which are made of a single cell, but they are still visible to us. Do try to find about them and how do they look like and how do they function. However, we will now just look at some of the images of the structures formed by the cell or the cells that are present around us. The first image is that of a filamentous fungal cells, also called as the hyphae. So hyphae are basically the structures formed by joining of the two fungal cells, two or more fungal cells, and they together form a meshwork called as the mycelium. This next image B is that of Treponema pallidum, the spiral-shaped bacteria that causes STD, syphilis. The next image is that of the three important blood cells found in animals, the red blood cells, the white blood cells, and that of the platelets. This particular image is that of the cells seen as in case of human beings, where the red blood cell lacks a nucleus. This next image is that of a organism that is found inside oceans and usually have a strong shell kind of structure around it. And the rest of the cytoplasmic structure is embedded within these structures. This is an example of a radiolurin. Often these organisms are responsible for your uh, seabed formation and other aspects. This is an example of a ciliate, which is stentor. It is attached to a substratum which is not visible here, and it uses a cilia to direct the food materials towards it, and it engulfs the food particles through the process of endocytosis. Here is shown one of the most remarkable events in sexually reproducing organism, where sperm fuses with the egg cell to form a single cell zygote, which will ultimately give rise to a embryo. Then, this is the structure of intestinal cells collected together with microvilli present all over them, which increases the surface area for absorption. This is the next image of the xylem tissue, and you can see the tubules through which the water molecules move upward, the spiral structure and the spaces between them, which allows the lateral movement of water and also ensuring that the water moves upward in a single direction. And last, the most important and very beautiful cells, that is neuron. So this particular neuron shows the branches, the dendrites around it, and these dendrites are responsible for collecting signals from the surrounding and transmitting the information to the subsequent cells. Now, cell theory suggests that all living organisms are made up of cells. Cells arise from pre-existing cells. All cells have the basic chemical composition. Cells are the basic unit of life. Hereditary information is passed from the cell. Energy flow occurs within cells. Now we understand them in more depth. For example, in the first point, all living organisms are made up of cells in the structural aspect, and the organism can be classified as unicellular or multicellular. Then, cells arise from pre-existing cells by the process of mitosis or meiosis. Then all cells are composed of various chemicals which are called as biomolecules. These biomolecules can be macromolecules that can be also in large size, such as your DNA and proteins. Then your cells form the basic unit of life. For example, your viruses are often not classified as a living entity because they don't contain cells, but rest of the other organisms, which include your archaea, bacteria, fungi, algae, animals, and plants, they all contain cells, whether they are of unicellular nature or multicellular, or prokaryotic or eukaryotic. They all contain DNA, even including viruses. The better term to say than DNA is the genetic information because certain organisms instead of DNA contains RNA as well. And the RNA can also be of different types. It could be double stranded or single stranded, as we will discuss, and we will go into depth in future sessions. Then energy flows throughout organism, and the energy currency in living organism is that of ATP, also called as adenosine triphosphate. And what is important is that this energy is constantly being utilized and generated by the sum total of all reaction, which is called as metabolism. Now let's understand how do we measure cells. You can see that it is very difficult to go to a microscopic scale and then take a ruler and try to measure cells because that is not possible. But what we can apply here is the information of what you learn in physics. That is the idea of how magnification works. So you understand that magnification is equal to, if I write the symbol of magnification as M, it is equal to the size of the image, also called as the height of the image, upon the size of the object, also called as the size height of the object. Now, if we measure the length of the particular object that we have as seen under the microscope, we will get what is called as the size or height of the image. And since our microscopes are made up of lenses, they have particular focal length, they have particular points at which they form images, as we have learned in ray optics, and through that also we can calculate magnification. So we have the idea of magnification as well, how many times the image is getting magnified. And if you will plug in the values, you can find the size of an organism. Okay, so through that we can calculate the size of an organism. An organism usually are in the range of micrometer, where 1 micrometer is equal to 10 raised to the power minus 6 meter, which is 1 millionth of a meter. So when you divide 1 meter by 1 million times, the smallest piece that you will get will be equivalent to 1 micrometer, and the cells range from 1 micrometer to 100 or even more micrometers, but they are within the range of micrometer. And also what we see is that usually the bacterial cells, the archaeal cells are usually smaller in size, followed by your protozoan cells. And then animal and plant cells are quite bigger and larger in size. Plant cells often been even longer and often showing the process of elongation during their cell growth. Then the structure that are found within cells, which are called as the biomolecules, these often range in the come in the range of nanometer, where 1 nanometer is equal to 1 billionth of a meter. So, which is equal to 10 raised to the power minus 3 and micrometer because it is 1,000th of a micrometer itself. So, you can imagine how small these are because you are, in this case, that value 10 is being multiplied 3 times because you are doing the cube of that particular number. So exponentially smaller these particular molecules are in comparison to your larger cells. So these are very small structures as such, and large amount of these molecular structures form your entire cell. And a lot of movement happens within these molecular structures also, as we have already understood in chemistry as well, that particles are capable of moving. And these movement can vary from particle to particle, and the kind of interaction that they show can depend on the nature of the particles, whether they are hydrophobic or they are hydrophilic or other aspects as we will go in depth in the case of chemistry. Now, another aspect that we need to understand is about the cell surface and their volume connection. Because that surface area and volume ratio ensures that even with smaller size of a cell, an overall organism is getting advantage in the case of multicellular organism, and also in the case of your unicellular organism, they ensure that Even for a very small size, they will try to ensure that they have structures which increases their overall surface area for the given volume so that they can utilize more resources around them. So in this scenario, that is only what is explained. You have been given three cubes whose entire one length of one side is 20 micrometer, but it is subdivided over time to form a multicellular kind of structure. So what we see is that the volume in either of these three cases is 8,000 micrometer cube only, because the volume is basically calculated by the cube of the side. So the total side is still 20 micrometer. The surface area, however, is changing because when we divide this cube into subparts, then there are new faces which are appearing. And we know that the surface area is calculated by 6a square. So, for example, in this case, what is happening, this sides to calculate the surface area is continuously reducing. And also the number of boxes that are getting generated, number of cells that are getting generated is increasing as well. Okay, so that way we have to take into that into account, and through that we will calculate the surface area for the first cube as 2400, for the next as 4800, and the last one as 24,000. So you can see how suddenly the number has jumped. Almost tenfold increase has happened from this individual cell to this multicellular kind of structure. And also see, if we divide this number of surface area by volume, we find that the ratio is also increasing. Okay, so through that we can understand that surface area to volume ratio is very important for organisms. That is the exact reason why we have alveoli in our body, or why we have uh, villi or microvilli in our body. All of these ensure that the surface area increases manifold, even for a very small uh, space. So even large amount of volumes can be accommodated within a very small space. Okay. So, as we already discussed about sizes and how this can be affected by the surface area. So, the idea about your dendrites or branch structure is also to increase the surface that is exposed to the surroundings, and that way they can gather more information from the surroundings. And we can also see that within a single organism also the sizes can differ largely in a very large extent. So you can see that sizes vary enormously in an animal itself when we compare a neutrophil, which is a WBC, with a neuron cell. So in the next section, we will talk about cell visualization, that how do we visualize cells. So I hope you could understand about what sizes are and what is the size in which we measure microorganisms, and you will be interested to learn more about microorganisms. So let's see you in the next session.